Hi guys, welcome to the discussion of Chapter 6, Expression of Biological Information, and in this video, we will focus on transcription. This is the learning objective of this lesson. At the end of the lesson, students should be able to explain transcription and the stages involved. These include initiation, elongation, and termination in the formation of mRNA strand from 5' to 3' end. Let's have a look at the enzymes involved in transcription. Since during transcription you will be using DNA as the template to synthesize your RNA polymer, therefore, the enzyme involved is called the RNA polymerase. Unlike DNA replication, transcription is a much simpler process. It only requires one enzyme, which is RNA polymerase. And unlike DNA replication that only occurs in the S phase of the cell cycle, transcription happens throughout the life of the cell. Transcription of certain genes will happen as the cell needs the product coded by the gene. If the cell no longer needs the product coded by a certain gene, transcription of that gene will be prevented. The gene is now said to be turned off. The regulation of turning on and turning off certain genes will be discussed in detail when we are learning about lac operon. Now let's get back to transcription. There are 3 stages to transcription. It begins with initiation, followed by elongation, and ends with termination. Now let's have a look at initiation. During initiation, RNA polymerase will bind to the promoter region of the gene, like this. Once bound to the promoter region of the gene, RNA polymerase will begin to separate or unwind the DNA strands. Now, the RNA polymerase can use the template strand as the template to synthesize the mRNA. As to here, initiation process is complete. During elongation, RNA polymerase will begin to add RNA nucleotides complementary to the template strand, like this. Since the template strand run from 3' to 5' end, therefore the new mRNA produced will be produced from 5' end to 3' end. As the time progresses, the RNA polymerase will move downstream of the gene, like this. As the enzyme moves downstream of the gene, the enzyme will unwind the DNA and use the template strand as the template to synthesize the mRNA. Hence, you can see that the mRNA, the red line, grows as the enzyme travels downstream of the gene. Only the DNA at the region of active transcription will be separated. Behind the region of active transcription, the DNA will rewind again. Termination happens when the enzyme RNA polymerase arrives at the terminator region of the gene, like this. When the RNA polymerase arrives at the terminator region of the gene, it will detach itself from the DNA. The DNA will rewind and the mRNA will be released. Hence, transcription process is now complete. The newly synthesized mRNA will leave the nucleus, in the case of eukaryotic cell, to be translated by the ribosome that is located in the cytoplasm.